Hello, this is Melissa Herman, and I will be going over our cancer protein phd3. Abstract. phd3 is a protein that acts as a suppressor against cancer cells by using oxygen-sensing pathways to determine the fate of the cell. We use a co-immunoprecipitation blot to determine interactions between phd3, HIF-alpha, and actin. We found there was an interaction between the proteins phd3 and HIF-2-alpha, and we did not find an interaction between actin and phd3. We know trypsin-treated cells suggest that trypsin breaks down other cells and causes a degraded shape. We found it was similar to phd3 as phd3 also has an ability to induce apoptosis and is able to damage other proteins. We know angiogenesis is related to phd3 because phd3 is a suppressor protein and it will suppress angiogenesis, which would lead to suppressed metastasis. phd3 is a suppressor protein. It's a prolyl hydroxylase domain protein and is present under hypoxic conditions. It works at the gene level, so it's able to influence transcription and translation. It's involved in cancer because it promotes neural apoptosis, which is the programmed death of cells. It will suppress the growth factors, angiogenesis, differentiation, and metastasis of tumors. And we know it has an interaction with another protein, HIF-2A. Regulation of cell fate. The formation of agrosome-like structures are mechanisms for oxygen-sensing pathways that determine the fate of the cell. We know phd3 has an ability to produce low levels of oxygen through the activation of HIF-2A, which we saw interacts with phd3 in our co-immunoblot. And we also know phd3 has the ability to degrade HIF-alpha with the help of VHL through ubiquination. Here's our co-immunoprecipitation blot. You can see the control column with the different bands, and then we have phd3 by itself with a band at around 27 kilodaltons. In the next column, we have phd3 interacting with HIF-2-alpha. In the fourth column, we have phd3 with no interaction to actin, and that's indicated by the dotted circle. To perform the co-immunoprecipitation blot for detection of phd3, we broke down the cells using lysis buffer. We centrifuged the sample. We incubated the solution, then loaded the samples into SCS gel to run SCS page. After that, we would transfer the gel and observe the band's presence. A positive result is when protein-protein interactions occur, and we saw that with the band in the phd3 plus HIF-2-alpha column. We received negative results in the phd3 and actin column, showing that there were no protein-protein interactions. This is phd3's relationship to trypsin. Trypsin is a protease enzyme that's able to break down other proteins, causing cells to look degraded and lose their cellular function. We know phd3 is a suppressor protein that will inhibit cell division during hypoxic conditions, and it will promote apoptosis for tumors and cell death. phd3 causes cells to look similar to trypsin-treated cells because of phd3's ability to induce cell apoptosis, which is similar to trypsin's ability to damage cell plasma membrane and induce cleaving. Angiogenesis is the secretion of growth factors that signal the formation of blood vessels toward a tumor. phd3 will suppress tumor growth by inhibiting cell division and decrease the possibility of angiogenesis. Metastasis will be suppressed by phd3's prevention of angiogenesis. And here we have a figure of angiogenesis on a tumor, and you can see the blood vessels feeding and nourishing the tumor. Thank you.